guys, welcome to Fiveable. I hope you guys are ready. We're going to talk about plasma membranes. We're going to talk about components of plasma membranes. We're going to talk about fluid mosaic, and we're going to do some practice problems tonight. So I know that's what everyone really wants. So let's go ahead and get started. So my name is Jessica Nadsum. Follow Think Fiveable for all the updates about merchandise. Those are important. They have giveaways for that. Notes, study tips, streams, all those awesome things. So make sure you're following those. If you're a student, you probably have social media. So that's my pitch. But let's go ahead and talk about what we're actually going to talk about today. So like I was saying a minute ago, we've got a lot to cover. We're going to introduce phospholipids. So in the last unit that we covered, we talked about lipids, but now we're going to talk about phospholipids, which are very special lipids. We're going to talk about other components of the cell membrane that are equally important. If we don't have them, we don't have cells. If we don't have cells, we don't have us. You'd just be a blob of jelly on the floor. I don't know about you, but I don't want to be a blob of jelly on the floor. I like being a person and having bones and separated organs and all my emergent properties. They're great. We're also going to talk about the fluid mosaic model. So I hope you are ready for that. We've got some little intro images here. Let me see if I can get my laser pointer working. With our phospholipid, that diagram, and then with our spinning cell, because it is excited to be here. So it's just spinning, spinning, spinning. You can see that membrane is the part that's swirling, so then you can see the parts that are inside the cell. Notice how it's a little bit clear. Interesting things. Let's keep going. So we're going to open up tonight right with phospholipids. What can you guys tell me about or not even phospholipids, but tell me what you know about lipids. What do you remember about lipids from the last unit? A characteristic of lipids. Tell me about them. Pretend I know nothing. Hydrophobic, very good, Elena. So these are hydrophobic. They do not like water. It's like spraying a cat with a spray bottle. It makes them very unhappy. Non-polar, very good. So those two terms go together. Hydrocarbon chains, yes, I love that. These are going to be hydrocarbon chains. And so that's going to be part of the reason for their non-polarity. They have an equal charge distribution because it's just carbon and hydrogen, and those are not electronegative atoms. They're very good at sharing. They learned how to share their electrons covalently without problems from a very early age. So they are non-polar. Whereas things like water has an oxygen who never learned how to share, and so it's greedy with its electrons. It's very electronegative, and hydrogen is left standing outside with no electron at all. And that's what makes it do what? What does hydrogen do on a water? What does hydrogen do on a water that is so important for the properties of water? What does hydrogen do on a water? It is so important. Hydrogen bonding, very good, Lynn. So water is going to do that hydrogen bonding as a result of not having that electron because oxygen took it and refused to share. But luckily, lipids are very good at sharing, and as a result, they are non-polar. They do not mix with water. If you remember that phrase, like dissolves like. So keep that one in mind. So we're going to look here really closely, though, at a specific form of lipid. So we've got the word lipid in here, but notice we've got this phospho. What atom are we talking about when we throw in that phospho? What atom are we looking at there? Phosphate, very good, Lynn. So we've thrown in this phosphate, and if you look here at this guy, I don't know if I can. Oh, nope, I can't do that. Sorry, I was trying to zoom in for y'all, but I cannot do this on this platform. So you're just gonna have to do like me and get your face really close to the screen. Hope you don't mind my face. But we've got this phosphate here, and if you notice, there's a teeny tiny negative sign on that oxygen. We also have not one, not two, not three, but 4 oxygens surrounding that phosphorus. And as I just went on a tangent about, oxygen is very electronegative. And so that is making this phosphate head very, very polar, like more polar than a polar bear. And so it's got this very unequal charge distribution. All this oxygen is hogging all of the electrons. And meanwhile, down here on the southern region, we have these hydrocarbon chains. These hydrocarbon chains are very neutral. They sing kumbaya together. around a campfire with a guitar. They are at peace, they are balanced because everyone shares their electrons and they're nice. They learned how to share. And so these tails, these fatty acid tails on our phospholipid are nonpolar. This is very, 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 very critical because it gives it one of the most important qualities, and that's the amphipathic quality. So it says here amphipathic. If you've ever heard the term ambidextrous, for those magical national treasures of people who are equally good with either their left hand or their right hand, my brother is like that, and it drives me nuts. I'm like, how do you do that? Because I try to write something with my left hand, and kindergartners make me look terrible. So, but amphi, from the Latin word ambi, meaning both. So amphipathic, it's got both polarities. It's got a polar end and a non-polar end, which seems at odds with each other because we know those two things don't mix, but they are able to bond right here. You've got those hydrophobic tails that are binding at the base of the phospholipid, where there's just enough charge distribution for everything to mellow out. And so as a result, it's still going to bind to this glycerol, because remember, that's what fatty acids bind to. They bind to those glycerols, and everything seems cool. You got the non-polar end, you got the polar end. But, oh, this example may go over y'all's head a little bit, because I was a child of the 90s, and I don't know if y'all ever saw 90s cartoons on Nickelodeon, but they had some interesting ones. There was this show called Cat Dog. And to be perfectly honest, the show always kind of weirded me out. I don't know if you've ever seen Cat Dog. Has anyone here ever heard of that show, Cat Dog? Maybe you've seen it in like a BuzzFeed article, Throwbacks for Millennials or something. It's a cat and a dog. Yeah, it's about what it sounds like. So you've literally got, I don't have a pen, so I can't show you this. It's an animal where it's got the cat's head on one side and the dog's head on the other. It's about as disturbing as you can imagine, but Nickelodeon went through a dark phase, I think. But my point there is, if you tried to have an animal that is part cat and part dog, when you think of that, they don't go together. They don't. Now, if you've got an adorable dog and cat at home who love each other, you should tell us all about them, because I'm sure they're precious and I love animals. But on this show, they had a cat and a dog, and they were the very stereotypical archetypes. They despised each other. And you would think here that the hydrophilic head and the hydrophobic tail would hate each other, but they actually have a really good relationship because they do something special with each other. They form these bilayers and these micelles. So what they do is they put all of the tails together, all of those non-polar tails, because remember, like mixes with like. So you've got a micelle here, all these tails, and they come together because that non-polarity mixes. They're happy that way. But then meanwhile, you've got this polar end, and they have a place to go too, because then they're all grouped on the opposite end. This is what we call a micelle. It's a single layer, and they form these like bubbles. I really do think of them as bubbles. They're kind of like bubbles because they're flexible and they're cute and they're squishy. But these micelles will automatically form. You can put them in a bottle and shake up the bottle, and the micelles will automatically, you can look at them under a microscope, they will go back to this formation because of that charge. It has nothing to do with a omnipotent view of the world knowing, oh no, we're gonna get shaken, we're gonna get all mixed up. It's just, that's their charge. They seek out having that equal distribution of charge all in the middle and having that unequal distribution of charge, the polar end, on the outside. They do that on purpose so they're protected. Now keep in mind here, I said this happens in aqueous solutions. What's an aqueous solution? A solution with water, exactly. And so water solution, thank you, Sandra Lynn, very good. So yeah, aqua, water, another Latin word, there you go. But it's an aqueous solution, it's a solution in water. So you're automatically gonna have these non-polar tails who are very unhappy about their circumstances, and they don't wanna touch water, they don't wanna be anywhere near it. Another reference, if you ever read Dr. Seuss books, if you remember the Sneetches, and they had stars on their bellies, and if you didn't have a star on your belly, they wouldn't talk to you. They were very rude about that. Phospholipids are the same way. If you are polar, then the hydrophobic end, the non-polar end, will not talk to you. And so it goes and clumps up like this. And that works out fine because that polar head is all grouped together and it forms a barrier. It protects them in these micelles. So that's the single layer version. But then the version that we actually use in our cells is this double layer. And so here the tails are still touching, but it forms these layers, like a double layered cake, because that sounds delicious right now. And so you've still got your tails on the inside, and they're protected from that aqueous solution. It can't touch it because the polar heads are buffering all that polarity. And so everyone's happy. Now that being said, why can't our cells just be made of micelles? Why couldn't we just put our cells right here in the center of the micelle? Why couldn't we put all that celly stuff, organelles and cytosol, in the center of the micelle? Why couldn't we do that? Why has it got to be a bilayer? It'll get clogged. Okay. So it would get clogged with what? What would clog that micelle that perhaps the tails would not like? Think of the organelles. Think of all the molecules floating around in a cell. Do you think they're all non-polar? Cytoplasm. So we know in the cytoplasm, we have tons and tons of stuff floating around in that cytoplasm. You've got organelles like mitochondria and nucleus and RNA, and you've just got random ions floating around too. And those ions have a charge. And sometimes they have an unequal distribution of a charge. So inside a cell, if I can go back here for a sec, inside a cell, you've got all this stuff floating around. Do you think all that stuff floating around inside the cell is non-polar? No, it's not. Very good, Lynn. So all that stuff floating around inside the cell is not non-polar. A lot of it's polar. And we can't do that because then the tails would freak out because they'd have something polar inside. It'd be like the Trojan horse getting into their defenses. So we have to use this bilayer because then even if I've got the inside of my cell and the outside of my cell, the polar heads handle any of that charge and they don't have to deal with it. And that's the way they like it. Non-polar tails don't want to touch anything with a charge, anything polar. They don't. They're very, very snooty and they must remain non-polar. They don't mix with others. Okay, so this is just kind of an example. You may have seen the commercials for this stuff on TV. Micellar water. This is the stuff they market. You know, the woman comes out and she's got like perfect hair and perfect makeup. And she's like, I use micellar water now because it's oil-free and blah, 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 and whatever else she says. And she does the thing where she like washes her face and splashes it, and somehow it doesn't mess up her hair or make a huge mess in her bathroom. And she comes up and she still has makeup on, you can tell, but they say, oh, her makeup's all come off. So the commercial's a little bit misleading and it's not very scientific. But I do have a point here. Micellar water is made of micelles. That's all it is. Keep in mind, it's got those polar heads. They pick up all that polar dirt and makeup, and they clean it out of your skin. I do actually use it every night. It is really handy, and it is really makes my skin feel good. This is not a scientific endorsement, nor is it a scientifically researched thing that it's better for you. But that's how it works. It has those micelles. You put it on a cotton pad, those micelles stick to it, and then they, with those polar heads on the phosphates, clean all the makeup off. I recommend using cotton. Please don't splash all the water on your face. You're just gonna make a mess in your bathroom. The floor is gonna get wet, you're gonna slip. It's not gonna be good. But that's how micellar water works. So now you see, science and practice, it is a real thing. Okay, so let's talk about the broader version of this, the actual plasma membrane, the thing that you actually showed up to see. So looking at the plasma membrane, hang on, I'm trying to make sure I didn't mistype anything because okay, no, it is correct. Sorry, the font's really, I have a really small computer screen, so I can't see some things. So I promise I'm gonna get classes someday. But so this plasma membrane, and I'm sure you've heard of cell membranes, you've talked about them at some point in your life because that's what you do when you go to Starbucks, you just sit and talk about cells. But The plasma membrane is flexible yet durable. So the thing I want you to do is grab your arm and twist it around. Just twist your skin, your fingers and stuff. Maybe pop your knuckles. Do this. Reach up for the sky. Just do some chair yoga. And I want you to notice that as you move around like this and do all these things, that as your heart rate rises, because I don't exercise enough, your skin doesn't break. I'm bendy. I can flex around. I can't like put my arm behind my head and then put my leg behind my head. I'm not that bendy, but that's a joint problem. That's not a skin problem. But my skin is very flexible. It moves with me, and I need it to. If it didn't, I'd be like a tree. I'd just have to sit there. Like, please let the sun shine on me. But we're not trees. We don't have cell walls. All we have is a cell membrane, and that cell membrane is very flexible, just like your skin. I like to think of the plasma membrane as literally being the skin of the cell. It's flexible. If I push it in, it bounces right back up. If it doesn't, you need to get that checked. But it's supposed to bounce right back up, does what it's supposed to, but it's durable at the same time. It's not like if I pinch it too hard, it's gonna break. I mean, I guess if I pinched it really hard, it would break, but for the most part, my skin's pretty durable. And so are your cells. And the reason for that is, so like I showed you back here, you've got these bilayers. Now, if you look at these tails and at these phosphates, they're not anchored together by anything. They're not like, okay, I'll tie one around me and you tie one around you and we'll all be connected in a nice chain. They're connected, but it's just their weak attractive forces. It's not anything major. They're not like cemented together or locking their elbows. And so as a result, they can flex and they can bend. And that's why your skin can bend, because you're made of cells and your cells are a little bit flexible, which is advantageous, like I said. But your cells can do this because the way that these are bonded together is loosely. And so I'm going to show you in a minute what that really looks like, and you're going to be like, that looks like an alien landscape. And I'm going to be like, nope, that's inside your body all over the place. But so this cell membrane, flexible but durable, but the main thing is it's the barrier that separates the cell from the outside world. So as a full-grown adult who works in a school and encounters germs constantly, get your flu shot, by the way, if you haven't already, but I am constantly encountering germs. I am constantly terrified. What is my body's biggest defense? What is the biggest organ in my body and the biggest component of my immune system? Skin. Very good, Elena. Skin is your body's biggest defense. People who have severe burns who pass away, the reason they don't survive isn't pain from losing their skin, which, as terrible as that sounds, the reason they don't survive is they have no immune system. All the bacteria and viruses and other things that are just hanging out on my skin and in my classroom can now get into my body very, very, very easily. You have to have skin, and you have to have cell membranes, because that barrier keeps all the bad stuff out of your cells while also at the same time letting through the good stuff. So for example, with my skin, there are certain things I can put on my skin that keep me healthy, like lotion, so my hands don't dry out, which they try to do frequently. But there are also certain medications that you can put on your skin. So for example, there's a really common one, it's called promethazine. And it's an anti-nausea medication that they give to people because when you're nauseous, you're throwing up, you can't really swallow pills because you throw them up. I hope you're not eating dinner, I apologize. But so often it's this topical medicine and they'll put it on your skin where it's really thin and you just rub it in and that makes you stop being nauseous. And so our skin does let in certain things that are good, and that's a really good thing. Your cells let in certain things that are good as well, like water, ions, food, hormones, all sorts of stuff. But they keep out the bad stuff for the most part. And the reason for that is, going back to this phospholipid bilayer structure, I've got these polar heads, I've got these nonpolar tails. Nothing can get past those nonpolar tails. that isn't nonpolar. Only nonpolar things get through here. Now, spoiler alert, the majority of nonpolar molecules are nonpolar because they're pretty large. They're big molecules and they don't fit through these teeny tiny gaps. There are some, most of which are gases like atmospheric oxygen, that just slide right through because they are nonpolar. But this is a perfect barrier. That amphipathicness, being both polar and nonpolar at the same time, is perfect because it keeps things from getting through unless it is specifically supposed to get through. So it's a perfect barrier. It's bouncy, so if you hit it, it's not just going to collapse. But it's durable enough that if something really does hit it, it says, nope, you shall not pass. And we like that. So the metaphor I always use, I think cells are a lot like bubbles, very flexy, very bouncy, not quite like bubbles because bubbles do pop. But in my class, we actually do a lab where we make bubbles out of corn syrup and soap. And those bubbles do not pop. They will sit on my desk for 3 weeks and not pop. And that's what most cells are like. They just sit there and they don't pop, but you can poke them and they just hang out and then they come back no matter what you do. So coming back to this whole idea of only certain stuff goes through, the stuff that is specifically supposed to go through. That's where we get to these things. They're called membrane proteins. So you can see I've got a little animation of it up here. You've got your bilayer with a layer of phospholipids on top, a layer of phospholipids on the bottom. And you've got this protein and he looks like he's just kind of grabbing stuff and throwing it out, grabbing stuff and throwing it out. That's a transport protein. He's doing exactly what he's supposed to do. He is taking something that I imagine this little red dot being very polar. And why wouldn't that red dot be able to get through the phospholipid bilayer without that protein? Why can't this red dot get through the phospholipid bilayer without that purple protein? What's a quality that would prevent it from crossing? It's polar. Very, very good, Elena. That little red dot, if it's polar, it would try to get through here. It would get to the heads, and then it would hit the tails, and the tails would say, "Nyeh!" And then it would get banished away. It wouldn't work. And so it has to have this protein that can transfer it through because the protein has to have a non-polar aspect to it. So these membrane proteins, they can be both hydrophilic, meaning they're polar, or they can be hydrophobic, meaning they're non-polar, or they can be both. Could be either one, because if you think about it, they've got to play nice with everyone. They've got to play nice with the polar phosphate heads. They've got to play nice with the non-polar fatty acid tails. Sorry, I've been up very early. But these things, we say they stud the sides of the membrane as a mosaic. And so just kind of popped into the membrane right here. And so they just take up a little bit of space and they do lots and lots of different things. But this is why you need your proteins and you need them to be very, very specific. So again, calling back to the last unit, because in biology, everything builds. It's all about the structure of that protein and what job it can do. It's all about the amino acids that make it polar or make it non-polar, because they can do so many different jobs and every single job needs a different structure. So for example, we can have this one right here, 2 proteins coming together to hold cells together. And some instances where we need cells to literally hold hands and hold on to each other and don't let go. They can be for enzymatic activity, meaning they can break things down or build them up, anabolism and catabolism. You're going to learn more about those later. But we're going to need those proteins to absorb things and break them down, or absorb them and build them together. And sitting in the membrane is a really good spot because it's going to be anchored there, it's going to sit right there, and things bounce off the membrane all the time. They're also going to be used for transport. So sometimes it's literally just going to be a tunnel, a way for stuff to get into the cell or a way for stuff to get out of the cell. And there's no toll, there's no one guarding it, there's not a troll under the bridge. It's just a hole in the cell that says, hey, if you need to get through, beep, beep, here you go. And that's great too. Sometimes it's going to be for cell-to-cell recognition. So you're going to have these little antennas sticking off, like your parents used to have in the 80s, before everyone just had cable and Netflix. And that antenna sends out little signals to the alien saying, hi, I'm here. And then all the other alien cells say, hello, I'm here too. Let's do cell stuff. And so you've got to have those as well. and they need to be able to go outside the cell to talk to the other cell. If it was just hanging out inside the cell, that wouldn't help anyone. They can also be for anchorage. So if you've ever built a house, I'm making a broad assumption, assuming people have built a house, but you should probably get the idea. You need a foundation when you build a house. Those proteins serve as the anchor or the foundation, so you can attach other things, whether those be proteins for signals or a cytoskeleton, whatever they are. And then finally, these proteins can also be used for signal transduction. So that means if we are sending a phosphorylating cascade of signals, you'll learn that later, and we want to trigger some sort of reaction like a hormone being released, that protein is going to receive the signal and then say, hey, dude, we got a signal, until the next protein next to it, there's a signal, until the next protein next to it, there's a signal, there's a next protein next to that, there's a signal, until it gets to the end point that says, oh, hey, there's a signal. I shall now release the signal. That's all it is. So now looking at this, what do y'all think that purple protein animation is? What kind of protein is that? Transport. Very good, Elena. That's going to be a transport protein. Okay. I just saw a question from, please correct me if I'm wrong, Shreya. So Shreya asks, does water always need an aquaporin to travel through a membrane? Yes. So again, going back to this structure, non-polar tails. Can water mix with anything that is non-polar? No. Very good, Sandra. It cannot mix with anything non-polar. And so as a result, pretend this red dot is water, and it goes, woohoo, here I am, I'm going to hydrate you. Boom. It hits the polar phosphate heads, and the phosphate heads are like, yay, let's party. But then if it tries to go any further, it's going to hit those non-polar tails, and that's like the phalanx. It's not getting through, and so it bounces right back out. And so yes, Shreya, that's a very good question. You will need an aquaporin to get water in and out of your cells. And that's going to be the primary method that they use. Keep in mind, you also get water through dehydration reactions, but you use it up on hydrolysis. So here's some give and take. The other thing here, sorry, there are lots of different things that are going to need very, very, very specific proteins. So remember, enzymatic activity. Enzymes and their substrates have very specific shapes. Water aquaporins are going to have very specific shapes. So is a lot of other stuff that needs to come into your cells. One thing you're probably going to talk about with your teacher is the sodium-potassium channel. Those are highly specific. Cell-to-cell recognition, those antennas. So think of it this way. You've probably complained before that nothing on TV, even though you have like 600 channels, 2 streaming services, and a room full of DVDs. Maybe that's just me. But we've all done that at least once in our life. You've got YouTube in front of you, and you're still like, I can't find anything to watch. That's with 5 million channels. Those cell-to-cell recognition, these ones, have like 5 trillion channels of signals they have to recognize. That's a lot of different proteins. And all of these are studded in the sides of your cell membrane. So you think of it as being these pretty little bubbles. And they are. They're cool. But they got a lot of junk in them. Okay. Describe the roles of each of the components of the cell membrane in maintaining the internal environment of the cell. What do I mean by the internal environment of a cell? What am I talking about with that? The internal environment. Homeostasis. Very, very good. What happens if your cells get too dehydrated? Concentration gradient. Yes, Elena, that's a good one too. But what happens if your cells get too dehydrated? They shrivel up. What happens if all of your cells shrivel up? That means you shrivel up. I'm not ready for that. I'm in my mid-20s and already I'm at the point where I find a wrinkle and I panic. So the idea of shriveling up is not cool. But if your body doesn't maintain homeostasis, your cells don't maintain homeostasis. What happens if your cell gets too hot? What if the temperature rises too much? Proteins break down, they denature very good. And then all of these proteins are suddenly the wrong shape and they can't do their job anymore. Maybe they can't bring in food, maybe they can't bring in water, and then the cell shrivels up. but that would be a problem. So the cool thing is components of your cell membrane maintain that internal environment. They prevent problems from happening. So one of those would be maintaining temperature. Your cells don't freeze, which if you think about it, it's kind of interesting because if you know most fats, phospholipids, most of those will freeze, especially if you think of like butter. What happens to butter when you put it in the fridge? A fat. It hardens. And then you try to make toast and you ruin your toast. And you're very sad because you only had 20 seconds for breakfast and now it's ruined. Not that this happens to me regularly. But those fats, when they're cold, they harden, they solidify. This happens to most fats. If you put olive oil in the fridge, it'll do this to a degree as well. It will congeal. Animal fat is solid when it gets cold. Your fat is solid when it's a certain temperature. But when you get cold, when you're exposed to cold, when you go stand outside and it's maybe a little bit chilly out that day, does your body start congealing? Do your cells get hard? Does your skin get less flexible? Yes or no? Does your skin get less flexible? No. Very good, Elena. Your skin, when you stand outside, you're freezing. Your nose is turning a little red. It's probably runny. And you're probably shaking and your teeth are probably chattering if you're me and you prefer warmer temperatures. But your cells are fine for the most part. And that's because they have roles of components to maintain homeostasis cholesterol. So in addition to having these proteins in your cell membranes, you also have cholesterol. And you may have heard of cholesterol. Maybe like your parents have talked about it, like, oh, my cholesterol's high. My dad has high cholesterol, and so he's always like, yep, it's high. The doctor switched my medication and da-da-da-da-da. And I'm like, that's nice, Dad. Take your pills, please. We would like you to live until you're 102 for your grandchildren. But my point on that is there is some cholesterol that's good for you. You have to have fat. If you don't, you won't. It's not good for a variety of reasons I don't need to get into here, but primarily because your cells need fat. That fat is insulation so that when you get too cold, you don't just shrivel up and harden and freeze. The fat insulates you. Think of a baby seal. We've all seen pictures of baby seals when they're perfectly white and they're adorable. And they're just grinning at you out of the snow. This is pictures. I've never seen a real baby seal, but you get the point. Why are baby seals so fluffy? What do they have? Blubber, which is fat. And they had to have it because those baby seals live in the middle of the Arctic Ocean and it's very, very cold there. And so that blubber insulates them the same way your cells insulate you. So if you step outside and it's not too warm and cozy outside, you don't freeze. So that's going to be really important in maintaining the internal environment of your cell. We're also going to have these proteins that, let's say there's not enough water, those aquaporins will let in more water. Or maybe there's too much and your cell is actually swelling. It'll take some of the water out. There's also going to be channels that move ions and keep your concentration a certain level. You ever had your blood sugar tested before? If that gets off too much, too high, or too low, that's not good. And so we have channels that make sure that glucose can get in and out of our blood and maintain a certain level. We have proteins for that. We have cholesterol for maintaining temperature. All of these are extremely important. I'm going to go ahead and move on to the alien landscape. So this looks like a straight up alien wasteland. I found it on Jiffy and I was like, this is perfect because this is the fluid mosaic model. And guys, I know the Mona Lisa is the best work of art ever, but for a biologist, this is a close second. So what's this beautiful orange line along the center of your screen? What is this beautiful orange barrier? What is that? The bilayer, the phospholipid bilayer. And do you notice how it's flexible? Like one of those little bounce houses that kids have where they're bouncing around and it's inflated, but if you fall down, then you might as well stay there because people are going to keep bouncing and you can't get back up. It's bouncy. It's doing exactly what it's supposed to. Which side is the inside of the cell, up or down? Which side is the inside of the cell? Down. Very good. What's that big thing in the center of the screen? What do y'all think this is? I talked about it in one of my last streams. What is that thing? Long mitochondria. Very good, Elena. That's the mitochondria. Okay. And so that was just, we hadn't learned that today. I was just curious. But notice this fluid mosaic. We say it's fluid because it's bouncy, it moves, it's flexible like a little fish. But it's a mosaic. If you've ever seen mosaic art, it is beautiful. It is where you have these tiles intricately glued together to create a beautiful picture from a bunch of little pieces put together to make one single picture. It's gorgeous artwork. But we say this is a mosaic because it's got lots of teeny tiny pieces built in to make this full image. So for example, I've got proteins here. I've got signaling molecules here. I've got all these molecules bouncing in and out. There's tons and tons of stuff going on. Now, do you notice how some of these molecules, like right here, they bounce into the bilayer, but they don't get through? Why aren't those molecules on the middle left able to get through? What's wrong with them? They're too large. What else could it be? They're hydrophobic? Well, keep in mind the tails are the hydrophobic part. So is it that they're hydrophobic or hydrophilic? If this stuff can't get through, it can't get through the middle of the bilayer. What's the middle of the bilayer? Are the molecules hydrophobic or are the tails hydrophobic? The tails are hydrophobic. Okay, thank you for confirming, Shreya. So yes, the bilayer is hydrophobic. It can't get through. It can't. It's polar, it's bouncing off. It's not gonna work. But this is your fluid mosaic model. Nothing will make your teacher happier than you being able to say the fluid mosaic model is the idea that the cell membrane is fluid because it is flexible, yet a mosaic because it is composed of many parts. It's literally all a biology teacher wants to hear after the mitochondria is the powerhouse of the cell. Non-polar stuff can pass through. So Elena just asked, can non-polar stuff pass through? Yes, non-polar things can pass through if they're small enough. So I'm assuming here that these molecules are either too large or that they're polar. I don't know that they're too large because if you look at these proteins hanging out in the middle of the bilayer, they're pretty big, bigger than these guys. So I don't know if it's size or their polarity, but that was a very good question, Elena. All right, we got a few minutes left. Let's get to the part that y'all all want to talk about. Let's do some practice problems. Practice AP question number 1. Which of the following statements is true regarding the movement of substances across cell membranes? Which of the following is true regarding the movement of substances across cell membranes? So we've got 4 options, so I want you to take like 30 seconds to read through those options. I'm not going to speak because I want you to focus. Take about 30 seconds. See a lot of A. Okay? Why do we say A? Ions are unable to move through the phospholipid bilayer because of the non-polar tail regions of the phospholipids. Okay, so I'm not going to go through all of the options, but what's wrong with option B? What's wrong with option B? Because option B talks about ions as well. The heads aren't charged. So let's take a look. Da, 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 da. A. Very, very good, guys. It is going to be A. Y'all ready for the next one? Here we go. All right, practice question number 2. Which of the following best describes the numbered areas? So again, just go like this and stare at your screen. We've got area 1, we've got area 2, we've got area 3. So again, I'm going to give you all like 30 seconds. I've got my watch timer. Whenever you're ready, go ahead and put your answer in. So A says areas 1 and 3 are polar since the membrane molecules are aligned with water molecules. What's wrong with C? Because it says it's hydrophilic. That's the same thing as polar. What's wrong with C? There is not a covalent bond with water. Very good, Elena. And the heads don't form covalent bonds. Very good. Yeah, they're way too electronegative for that. So it's going to be A. Very, very good. Y'all are 2 for 2. Let's keep going. Okay. The model below shows the structure of a portion of a plasma membrane. So we've got our plasma membrane here. It kind of looks like it's opening like Baby Shark style. Plasma membrane in an animal cell. Which statement best explains the orientation of the phospholipid molecules in this model? All right. Once again, 30 seconds. Go. Take about 5, 4, 3, 2. B. You say the hydrophilic phosphate groups of the phospholipid molecules are attracted to the aqueous internal and external environments. Remind me, what does aqueous mean? Water. Very good. Very, very good. Okay. I don't have an alternative here, so I'm just going to go ahead and tell y'all. Woo! 3 for 3. Y'all are killing it. Let's take a look at the last multiple choice question. All right. A model of the plasma membrane showing several biological molecules, including a transmembrane protein, is shown in the figure. Which statement best explains why correct protein folding is critical in the transmembrane protein shown below? All right, y'all. This is bringing it back to the last unit. Why does the protein have to fold properly for this to work? Interactions of the hydrophobic and hydrophilic amino acids help to anchor the protein. Okay. So what makes y'all pick A instead of B, C, or D? How did you rule out B? What's wrong with B? Peptide bonds don't interact. Very good, Elena. Peptide bonds are solely between the amine groups and the carboxyl. Perfect. Good job, Shreya, with the membrane. Very good. And then C, it says interactions of the protein and phospholipids increase membrane fluidity. D says interactions of the quaternary structure will increase hydrogen bonding. Nope, nope, nope. So very, very good, guys. Ta-da! 4 for 4. If I could give you a 4 right now, I wouldn't, because I would give you a 5. Y'all rock. All right, I got one more for you. I'm going to warn you, this problem's old, like 2 modifications old. But your teacher will probably forgive me for that because she doesn't want me to give you the current ones that she's going to give you on her test. Or he. I don't mean to assume your teacher is a he or she. But so this is a practice AP FRQ prompt. And like I said, it's like a decade old. But it's got the key points. Identify, discuss. What are the 3 macromolecules that are components of the plasma membrane? So first off, I just want you to identify them for me. What are those 3 macromolecules? Lipids, proteins, carbohydrates. Okay, good. What are the lipids doing? What are those lipids doing? Insulation, bilayer, very good. What are the proteins doing? Everything, yes. Transfer material. Elena, I love everything because, as I tell my kids all the time, it's true. But transfer material is slightly more specific. What's another example that we saw? Not just transportation, but signaling, communication. Very, very good. And what are we going to use those carbohydrates for? ID flag, like identification flag? Yeah, they're going to be involved in some of that identification. Very, very good, Elena. Great job. So I'm going to go ahead and show y'all. This is really teeny tiny, so I don't know how well y'all can see it. So little bit of warning here. You would want to get real specific when talking about these. So lipids, proteins, yes. But with that carbohydrate, they would have wanted you to say something pretty specific like glycolipid, glycoprotein. We haven't talked about that specifically today. That gets more into next stream, I believe. But I think you would have gotten partial credit because this is one where you can get 6 whole points. A point for each of these. So, woo, good job. Y'all rocked it though. Y'all are doing so good today. We did it. So guys, I'm real sorry. I don't have like more planned. But we covered it and y'all did awesome. So I'm a little early, but I don't have anything else. And from the looks of those practice questions, y'all get this. So I'm gonna go ahead and say y'all have earned 9 minutes of extra free time. Slides. I want that chip. Yes. Okay, so these are going to be posted on the Fiveable website. Go ahead, give me just a sec. And you have definitely earned the slides. So let me get that link for you. And I'm gonna stop sharing here because that's a bunch of my screen. You don't need that. Slides. You've definitely earned yourself some slides. So, ta-da! This will obviously be on the Phi Bubble website, so if you don't have this link later, it'll be up there by tomorrow. Full disclosure, my computer's pretty old, and so downloading takes a while. So I usually just have it download overnight. But like I said, yeah, you guys have been awesome. If you have any questions, feel free to tweet at me or something, I don't know. So, whoop, whoop. See you next Thursday, y'all. Peace out.